Hello, my name is Tracy. I'm an R&D scientist at Standard BioTools, and I'm really happy to be speaking to you today about our work on a multimodal approach to mapping anti-tumor responses in whole blood and tissue biopsies of cancer patients with our mass cytometry platform. So immunotherapy is really a revolutionary class of treatments that has had limited efficacy in solid tumors. Now, in clear cell renal cell carcinoma, or CCRCC, this is the most common subtype of kidney cancer, reliable biomarkers that predict who will benefit from immunotherapy and which treatment will be most effective are still lacking. Now, there are many contributing factors to this, including a heterogeneous tumor microenvironments, undefined immune cell, tumor cell interactions, an incomplete view and understanding of the functional diversity present of immune cells, and barriers to immune infiltration, to name a few. Now, in order to predict and improve outcomes, we really need to look at tumor biology from many angles. High levels, sort of pan-tumor microenvironment angles, single cell functional profiling. We need to be doing assessment of both the local and systemic immune systems. And to do this, we really need to use more than one technology. We really need a multimodal approach to address these important questions. Now, this is where mass cytometry comes in. So we can see all of these different facets of tumor biology with one technology on one instrument. So to study this in CCRCC, we obtained matched tumor and whole blood samples from stage 1 and stage 3 patients. So first we used fixed tissue slides to stain with our metal tagged IMC antibody panel. And then we ran samples on the Hyperion XTI, which is our imaging mode, and performed quantitative single cell spatial analysis. In parallel, we obtained dissociated tumor cells from the same tumor and matched peripheral blood mononuclear cells, or PBMC, for each patient, which we stained with our metal tag CyTOF antibody panel. And we ran these samples on the CyTOF XT instrument, which is our suspension mode, and then performed high-dimensional single-cell functional profiling. And then finally, we consolidated these two data sets to obtain integrated insights. And today I'll be showing you data from the stage 3 patient in this talk. So here's an overview of the antibody panels we used in this study. We design and optimize these panels in just a few weeks. And on the left, you see the 43 marker CyTOF panel. On the right, we have the 41 marker IMC panel. And in the middle, you can see this overlap. These are the shared markers between the CyTOF and IMC panels. And this really helps us integrate these two data sets together. Now, in both of these high parameter panels, we are measuring intracellular markers, including transcription factors and cytokines, in the same cell as our surface phenotyping and functional markers. And so with these panels, we can look at tumor cells, stromal cells, endothelial cells, extracellular matrix components, over 20 immune cell subsets, and more than 23 functional readouts. So in this study, we wanted to address three main questions. What is the spatial architecture of stage 3 CCRCC tumors, and can tissue heterogeneity be resolved? At the single cell level, what is the functional profile of immune cells? How does this differ between local and systemic immune environments? And third, what is the spatial relationship of key functional populations we can identify to other cells within the tumor microenvironment? So we'll start with our first question, trying to look at spatial architecture and tissue heterogeneity. So we first examined a high-level view of the entire tumor tissue section. So we can visualize here with IMC tumor cells, stromal cells, endothelial cells, and lymphocytes. Now, clearly the tissue architecture is heterogeneous. We can see that both tumor and stromal regions show accumulation of lymphocytes, visualized here in red, at two different sites of immune cell hubs. And we can see the relationship to, for example, blood vessels, which are shown in these zoomed in images as green staining cells. To determine the phenotype of these immune cells, we can visualize specific markers of lymphoid and myeloid lineages. So we can see in this region, just outside of the tumor margin here, we have a large hub of mostly T and dendritic cells. We can also visualize tertiary lymphoid-like structures, which form within the tumor and the stroma. So what you can appreciate from this lower zoomed in region here is that we have B cells in sort of the core of the TLS. We have T cells and dendritic cells also present. And in both cases of these immune cell clusters, we can see macrophages, which are shown in red here, sort of surrounding these structures, but not actually penetrating into them. In addition, we can analyze functional readouts, for example, of the mTOR signaling pathway using our phospho-4 EBP1 and phospho-S6 antibodies to identify their expression. So interestingly, we can see these two distinct branches of the mTOR signaling pathway are active in a mutually exclusive fashion at these two different immune cell hubs. So one within the tumor mass and one within the tumor stroma, demonstrating heterogeneity of active signaling pathways across the tissue. So then we took all 41 protein signals and we performed pixel clustering analysis to map clusters of cells throughout the tissue. So doing this, we can visualize areas containing necrotic cells, tumor cells, stroma cells. We also get a detailed view of the organization of immune cells within the tissue. So we see these TLSs, both within the tumor area and outside of it. We can also see areas of immune infiltration shown in blue here. And interestingly, we see a very distinct region on the bottom left here in yellow of these TIM3 positive tumor cells in a tumor region that is cold or lacking immune cell infiltration, suggesting there's some sort of mechanism of immune evasion taking place here. So after obtaining this high-level view of tissue architecture, we next wanted to go deeper into the single cell composition of the tumor and look at cell functionality with the more than 20 functional markers in our CyTOF panel. So we took part of the same tumor that I just showed you, and we obtained dissociated tumor cells and then ran our 43 marker CyTOF panel. And we phenotyped all the major cell populations, which are listed here on the right and visualized in this OPSNI map where each dot represents a single cell. Then we looked at functional marker expression within each of these cell subsets. So this heat map here is showing the median intensity of each marker on the y-axis within each subset on the x-axis. Now we also had access to a matched peripheral blood sample from this patient, so we could run the same analysis and compare local versus systemic immune functionality at the single cell level. So here I'm showing you the same heat map, but for PBMC. And so as an example of some of the interesting findings that came from this analysis, we found that non-classical monocytes appear highly functional in PBMC, expressing PD-L1 and TGF-beta. However, differentiated macrophages in the tumor microenvironment, or TDC sample here, become dysfunctional and express things like TOX and TBET. Now, because of the importance of T cells and antitumor immunity, we dug deeper into the CD3 positive compartment and we quantified T cell subsets at a more granular level. So what you can appreciate is that there is a large increase in the CD4 positive memory subsets that we see in tumor-derived cells compared with peripheral blood. In contrast, we see more naive and cytotoxic cells present in the periphery compared to tumor-derived cells. So we next assess the expression of functional markers in these T cell subsets. For the sake of time, I'm going to focus on just a couple of the interesting phenotypes we observed in CD4 memory cells. But I hope you can appreciate just how many functional protein readouts we can assess at the same time and see the sort of striking diversity that we can really only detect using this many markers at once in a single cell. So here we can see in the CD4 memory subset a much higher number of Tregs in the tumor-derived cells compared to PBMC. We also see a lot of exhaustion markers being expressed in these cells, so highlighted here in the purple box in the heat map. And then as an example in the bivariate plot, we can see high levels of PD-1 and CTLA-4 in TDCs compared to the PBMC. So overall, we're seeing more naive and cytotoxic effector cells in the peripheral blood and more memory cells with an exhausted phenotype in our tumor-derived cells. So finally, we had this exciting opportunity to integrate these two data sets together and try to map the populations of interest that we profiled with CyTOF within the tumor microenvironment with IMC to ask what is the spatial relationship of these key functional populations to other cells within the tumor microenvironment. And the way that we do this is by first taking a lower resolution preview mode image of the entire tissue section. So this 12 by 13 millimeter section was imaged in about 20 minutes with over 40 markers simultaneously detected. So this is the fastest 40 protein simultaneous imaging technology out there. Now from this image, we can select our regions of interest and then run high resolution cell mode imaging. So you can see here this tertiary lymphoid structure, or TLS, very prominently here. And then what we do is we can apply our single cell segmentation analysis. And what this does is it isolates single cells. And from this, we can perform quantitative cell clustering to actually cluster these single cells. So when we zoom in on this TLS region, we can visualize how these cell types interact with each other. So we can also use the common markers between our CyTOF and IMC panels to map, for example, where these suppressive macrophages and exhausted CD4 T cells that we identified with CyTOF, where they are in situ in the tumor itself. Now in this TLS, we do see some PD-1 positive CD4 cells. So here they're shown in white, but most of the CD4 cells rather are PD-1 negative, which are shown in green. We can clearly see these M2 macrophages in blue that are encircling the TLS, but not penetrating to the core of the TLS. Now, if we look at another region of interest, which contains both tumor stroma and tumor cells within this, with this sort of large hub of immune cells at the top, right at the tumor margin, we can see numerous types of T cells, both CD4 and CD8 in green and red respectively, and quite a few PD-1 positive CD4 cells here in white again, right along that tumor border region. And again, the blue M2 macrophages are surrounding this immune hub, primarily within the tumor bulk, but are not found within this hub. So overall, using this quantitative single cell analysis approach, we can visualize the localization patterns of these functional cell types. In this case, we demonstrated that although the M2 macrophages and the PD-1 positive T cells are mostly non-overlapping, there's an interface at the tumor margin where they are right adjacent to each other. So in conclusion, I showed you how combining both CyTOF and IMC technology from matched peripheral blood and tissue samples offers a really comprehensive view of CCRCC biology by linking phenotype and function with spatial context. Now, these are insights that you're getting from the same instrument in two different modes. So the next stage of this work is really translating all of these interesting features that we identified into a larger study where we can use advanced data analytics to identify predictive features of patient outcomes. And this is something that mass cytometry data is really well suited for, and others have already published on this, which ultimately we hope will provide a path to improving patient outcomes. So with that, I want to thank all of the brilliant people at Standard BioTools who worked on this really exciting project. And thank you all so much for your attention.